Okay, so it's a pleasure to participate in this colloquium and to get to enjoy talks from such a great diversity of speakers from different disciplines. I mean, that's a really unique feature of this colloquium, and it's been a pleasure. I was given the title to speak about the coevolution of the brain, mind, and culture, but I have to give you full disclosure at the outset of this talk. I'm merely a biological anthropologist and a comparative neuroanatomist, so I dropped mind from the title. And to be honest with you, I'm probably only really going to be talking about coevolution of the human neocortex and its possible connections to social learning, as it might be relevant to culture. And that's because my job description as a comparative neuroanatomist and biological anthropologist is really to probe the details of how the human brain, as its machinery, is put together differently than the brains of our close relatives, and how it's evolved a different anatomical phenotype, which then might help us to explain some of the very distinct cognitive and behavioral specializations that we as a species exhibit. So I'll be situating my talk in the context of the evolution of culture as a very important human behavioral specialization, and then review recent research from other laboratories as well as my own to try to understand what has changed in the brain that might be related. Okay, so along with language, perhaps one of the most dramatically different things about humans as compared to other species, including other primates, is our capacity to generate, innovate, and reproduce culture. The definition of culture that's up here is from an American cultural anthropologist, which describes it as the total socially acquired lifeways or lifestyles of a group of people. And this is familiar to us. It's the varied manners of beliefs, customs, food preferences, dress, music, that are all passed down through the generations, not by genes, through observation, imitation, and social learning mechanisms that are so diversified in what humans do. And having a very well elaborated capacity for social learning leads to all these regional differences in behavior. But this is not unique in humans, of course. And as an anthropologist, a biological anthropologist, I think it's important to look to our close relatives for evolutionary continuity. And so it's well known that humans are closely related to the other African great apes. This is a recent estimate of divergence times, calibrated as well by fossil evidence, and showing that humans have their closest relatives with chimpanzees and bonobos diverging approximately 8 million years ago. And then next out in close phylogenetic relationship are the gorillas, and the next orangutans, et cetera. So it's important for us to look at evidence of rudimentary cultural variation in behavior in these other species to try to determine whether it is in fact so unique in humans. And of course, it's been well known for a long time that indeed other great apes show quite a lot of diversity in behavior across regions where they're found, local traditions. Some people do use the term culture to describe this kind of regional variation in behavior. And so this is a well-known diagram from Whiten's paper in 1999 showing with different icons different regional traditions and behavior ranging from tool use traditions and technologies to grooming postures that are used in different communities across equatorial Africa by different chimpanzee communities. And now several dozen different variants of behavior have been shown. Fewer variants have been shown among the different species of orangutans, and in fact even fewer in gorillas, but our closest, one of our closest relatives, chimpanzees, show among the greatest regional variation or cultural differences in behavior of any other animal that's ever been described. In a recent review paper, Whiten tried to characterize, and it looks like the top is cut off a little bit, tried to characterize the differences in cultural behavior that distinguish humans from chimpanzees and other great apes. So indeed, there are lots of shared similarities, but what is different? And so that's what's found in the right-hand column in this image. And I'm not going to go through all of this, of course, but important things to point out are that there's intentional teaching in humans. There's over-imitation that's been observed in human children, where the details of a task that are not necessary to accomplish the goal are imitated, whereas in chimpanzees, they'll find a more expedient means to solve a task, for example, with a puzzle box. And greater tolerance of strangers, and perhaps, as we discussed earlier, evidence of enhanced empathy and cooperation, which is all part of this package of social cognition in humans that is part of what contributes to our capacity for culture. So what I really want to focus on then is trying to discern, if we can, what might be different about the human brain that underlies our distinctive mental machinery, including culture in this case. As long as people have been comparing our brains to those of other animals, we have noticed that compared to our body size, we have extraordinarily large brains. And this is something that did not escape Darwin's notice, of course, among many others. And so this is something that is an unavoidable, important biological specialization of human neuroanatomy. We have brains that are roughly 3 times larger than one would expect for a primate of our body size, which means that our brains are about 3-fold larger, or 3 to 4-fold larger than what you see in the other great apes. And you can trace this change by looking at fossil crania in the paleoanthropological record and see that most of this brain size expansion has happened relatively gradually over time, progressively, over the last 3 million years, not with really any clear linkage to any tool technology innovations or anything else that can be seen clearly. So, fine, we have very large brains, but is this really sufficient to explain the very particular kinds of cognitive specializations that we exhibit as a species? Does brain size on its own explain things like language or culture? I don't think it does. So when you start to look at some more of the details about what goes along with our large brain size and how it's put together, we can obtain a little bit more insight. So one of the most disproportionately enlarged parts of the brain as it expands across vertebrates is the forebrain, and in mammals in particular, it can be seen as expansion of the neocortex. And what you see in this image, comparing a human and a chimpanzee, is that the neocortex is what's observed as the outer wrinkled surface of the brain. It comprises about 80% of brain volume in modern humans, and that includes the gray matter that contains the cell bodies, as well as the white matter. It's involved in the most highly integrative functions of the brain. But besides being so large, what else is special about the human neocortex that might help explain our capacity for social learning and culture? Has the neocortex enlarged uniformly, or have some particular regions differentially expanded or magnified to subserve the very particular kinds of cognitive behaviors that we're capable of? So these are important questions. And the insular cortex is a good place to start. So I'm going to talk now a bit about research that we've done recently looking at how subdivisions, or regions of the insular cortex, change in size across primate phylogeny and humans' place in nature in insular cortex organization. It's a good place to start because in this region, which is found here, buried underneath the sylvian fissure in humans, overhung by the frontal parietal and temporal lobes, there is this cortical area that has regions that are very important for social cognition, and they can be well defined. So there's a gradient of function across the insula from posterior to anterior. In more posterior regions, which are adjacent to secondary somatosensory cortex, you see a certain type of architecture which is well conserved and homologous across primates at least, where it's more granular cortex, and that means that layer 4 is very expanded, the thalamic input layer, and it occurs in the posterior dorsal portion of the insular cortex. And human functional neuroimaging studies indicate that this region displays activation and processing of body states and visceral body states like the sensation of temperature, pain, hunger, thirst, and sensual touch. But as you move more anteriorly within the insular cortex through the mid-insular cortex, which is defined as disgranular cortex, across primates where that layer 4 of small granular cells is not as well defined, and then on into anterior agranular insular cortex, these visceral body state representations get redeployed, reused. This is an idea that has been proposed by Bud Craig in reference to understanding emotional states and integrated with emotional information. So for example, the sensation of pain in one's own body activates the posterior insula, whereas empathy for other individuals' pain activates the anterior insula. It's as though using a reference to one's own body state information is useful in understanding other individuals' emotions. And so the anterior insula, here indicated by green, is important and has been activated in a range of different tasks that employ empathy, prosociality, but also things like disgust, shame, etc. So it's also important to point out we can understand a little bit more about the function of the anterior agranular insula by looking at pathology. So behavioral variant frontotemporal dementia selectively destroys anterior insula. And the symptoms that are observed in patients of behavioral variant FTD include inconsideration of other individuals' emotions. They'll be very rude and inconsiderate to their spouses who they've loved for a long time. They'll engage in disgusting behavior with absolutely no shame. They'll pick up half-drunk Coke bottles out of the trash and drink them on camera with no shame whatsoever. They'll sing karaoke terribly, look at themselves on video afterwards and think that they're doing great. So they have a very selective impairment of these social emotions. And in apes and humans, and also in other very large brain social mammals, the anterior agranular insula, and this is a close-up in humans of the most frontal part, frontoinsular cortex, seems to have these other neuronal specializations. In layer 5, these large projection neurons called von Economo neurons, which look very different than the typical pyramidal projection cell. And these are also very selectively destroyed in frontotemporal dementia, and they might be an anatomical specialization to help convey the outputs of this region in large brain social mammals. Okay, so the research that we've recently done in my lab is to look very descriptively at how changes in the size of these different subdivisions of the insular cortex change in primates and then how they might be related to the demands of social cognition. And this is research that's been led by a PhD student, Amy Barnfind. And using the histological, cytoarchitectonic homology of these 3 subdivisions of the insular cortex, which we can recognize across many different primate brains, we can then look at archival slide sections and subdivide them going anterior to posterior into these 3 subareas, here color-coded, moving from more frontal to more posterior coronal sections of the insular cortex. And we can do this through equidistant series of sections, integrate the surface area and get the volumes. So this is very standard volumetric approach in comparative neuroanatomy. And we did this for 30 different species of primates that were available. I should point out that the availability of these brains is mostly from zoo and biomedical facilities. So that's a limitation. We don't have wild living primate brains for these sorts of studies. And so what you see here is, it's too much information. I'm going to summarize it in the next slide. But the sort of data that we obtain across these different regions of the insular cortex plotted against brain size. And you can look at the changes in these subregions versus brain size across all these different primate species, use phylogenetic statistical methods to control for the effects of similarities just based on phylogeny versus what is independently evolved as a specialization. But to summarize those sorts of very complicated results, when you look at the scaling of insular cortex region size against overall brain size, what we see is this. The more posterior, relatively simple processing viscerosensory insular cortex scales against overall brain size with a allometric exponent very close to 1. And what that means is that it's close to isometry, which means that there is equal proportion scaling up. As the brain gets bigger, it gets roughly the same proportion of posterior insular cortex as it gets larger. But as we move more and more anterior from this mid-disgranular insular cortex and then finally to agranular insular cortex, the scaling exponent increases across primates. So this means that part of an increasing brain size, a byproduct that's expected is a disproportionately larger and larger and larger anterior agranular insular cortex, which in humans we know is very important in social cognition. Where do humans fall on this line? Do we have more than we would expect for a primate of our brain size, less, or just the right amount? These are the human points plotted on the non-human primate prediction lines. We have essentially exactly the amount of insular cortex subdivisions that we should have for our brain size. So even if we have more mental machinery that may be magnified and dedicated to this aspect of social cognition, it really comes as just a part of brain enlargement, part of the continuity or gradual change in brain organization that we would expect. So another question that we can ask is, with these data, is whether any interspecific variation from predictions for insular cortex size of agranular insular cortex is associated with social cognition capacity. And it's very hard to obtain a simple metric of social cognition capacity across many species, but a rough and ready measure that has often been used is social group size. So if we take the relative size of agranular insular cortex and correlate it with social group size across these primates using phylogenetic least squares multiple regression, where we can control for overall body size, we see that there is in fact a very good correlation between the relative size of agranular insular cortex and social group size, suggesting that as this part of the brain gets larger compared to the body, that it's correlated with perhaps some measure of increased social complexity. However, when we control for overall brain size, most primates fall so close on the line of prediction that there's no additional explanatory power once you factor out brain size. So much of this additional machinery in the agranular insular cortex for social cognition just comes with overall brain size. So that's some evidence from comparing adult brains and the structure of these parts that may be involved in at least some aspects of empathy and cooperation, which facilitate culture in humans. But I would argue that only comparing adult phenotypes in an attempt to understand the comparative neurobiology of culture is very, very, very incomplete because, of course, cultural capacities unfold over the course of development. It's the interaction of how the brain develops within an enriched social environment that results in the kinds of cultural demonstrations that we exhibit as humans. So now what I want to turn my attention to is talking about how neocortical development in humans may differ from apes. And I can tell you at the outset that we are at a point in the science where we have surprisingly very, very little information on what I think is a critical question. How our brains, how our neocortex develops differently from what is seen in our close relatives, the great apes. And without having this information, we can't know what is distinct about how our brain develops and therefore turns into the type of neural architecture that is culturally capable. And so to provide some justification for this argument that we have to look at the distinctive nature of human brain development, I'm going to piggyback on a publication from an earlier episode of the Colloquium in the Light of Evolution series, and this is from Boyd in his publication in PNAS, and I'm not going to read through all of this, but emphasizing the importance, beginning early in human ontogeny, our psychology allows us to learn from others, powerfully and unconsciously motivates us to do so, and shapes the kinds of traits that evolve. And comparative psychologists have pointed to the ways in which human cognitive development is distinct from apes. So this is from Tomasello and colleague, in pointing out that theory of mind, our understanding and ability to coordinate with others as intentional agents allows the mastery of linguistic communication as a scaffold very early on, within the first year of life, relying importantly on the recruitment of joint attention, which some people argue is especially elaborated in humans. And then by age 4, these psychological mechanisms are built upon even further to improve our understanding of the mental states of others, etc. We know that raising an ape in a human-like environment, highly enculturated, even with language training, providing as similar social stimulation as possible, does not result in a fully human-like behavioral phenotype. So there must be some interaction that we need to understand between how the human brain develops differently. So to make this point, before I present the evidence about how the human brain develops differently, I just want to emphasize that we know that neurodevelopmental timing, even in humans, variation between individuals is important in regulating cognitive function. And so this is from the research of Shaw, showing that in this image, you see the changes in cortical thickness over development from a longitudinal structural MRI study, and how it correlates this dynamic change in cortical thickness, the initial expansion and then thinning, how it is associated with a composite measure of intelligence, which in this case included causal reasoning and some other measures of general intelligence. And what you can see is that the individuals of higher intelligence, in this case who attained higher intelligence later, showed a more protracted, delayed developmental onset of the overproduction and then pruning of cortical gray matter material. And this is most selectively evident in parts of the superior prefrontal cortex. So the time course of development of cortical thinning is associated with later intellectual ability, and this variation is evident even in humans. It's also important to point out before I review the evidence of how human and ape brains are developing differently, that not only is the neurobiology different, but the social context of normal brain development in these two different species is also incredibly different. In chimpanzees and other great apes, the infant grows up very closely bonded to the mother. Is very closely physically associated, is in close proximity, and is not handed off for allomothering and care and support very often to other individuals. So the social interactions of an infant chimpanzee within the first year or two are very, very limited compared to what we see in humans. Humans much more regularly with their infants engage in allomaternal care, where older siblings help to care for infants, grandparents help to care for infants, even non-kin participate in the care. And in the context of an evolving psychology where joint attention is coming online within the first year of life, this may be very, very significant, that human infants within this period are engaging in interactions with a much greater diversity of social participants than just the mother. And then there's also evidence that cognitive development is regulated by the social environment. And this is well-known sort of data that either enrichment or deprivation can affect later cognitive development. And this is just one example of this effect. This is from a study by Russell et al. Recently, comparing enculturated bonobos and chimpanzees to standard laboratory-reared bonobos and chimpanzees, age and sex matched. And so these enculturated bonobos and chimpanzees are very well-known subjects from the Language Research Center that have received intensive language training. Some of them acquiring language competency, some of them not, but nonetheless receiving very intensive interaction with human caregivers. And the comparison sample are standard reared chimps and bonobos who are either nursery reared or reared by their mother. And they were tested on the primate cognitive test battery, which is a range of different tasks encompassing both social cognition and physical cognition, tasks that are very, very quick to deliver, but are large in number, including rudimentary theory of mind tasks, imitation, etc. And the important point here, as you can see, is that there is a major effect of enculturation in both domains, in social cognition and physical cognition, but probably most pronounced in social cognition for these enculturated chimpanzees and bonobos. So this argues that there can be a very important effect to boost social cognition depending on the social environment, even in chimpanzees and bonobos, but obviously something more is happening in humans. So ultimately, to understand the differences in human cognitive development, we need to better understand what might be distinct about how our brains develop, and this is where I'm headed. So if we just look at the raw data, comparing humans and chimpanzees, around the time of birth, human brains are already about 2 times larger than chimpanzee brains, having undergone more neurogenesis in fetal life. There's evidence to show from ultrasound studies that, in fact, a human brain grows at an accelerated rate in fetal life. And that's because the human brain achieves this very large size with only an extra 4 to 5 weeks of gestational time. So gestation is not tremendously longer in humans compared to chimpanzees. Yet the human brain has much longer to go postnatally to achieve its very large adult size as compared to chimpanzees. So what that means is that the proportion of adult brain size at birth is relatively smaller in humans than chimps. So there's going to be more postnatal brain growth to occur in humans before it's fully wired up and mature. And this is going to occur under conditions of being more developmentally plastic, as that increased brain size is achieved through increasing white matter accumulation, in fact, mostly white matter accumulation, and then modifications and additions to synapses and dendritic arbors. And this is going to occur for a greater proportion of maturation in humans in a more rich social context. And then it takes a little bit longer for humans to reach adult-like brain size. So the period of growth is a bit more prolonged. But how do humans get to have that large brain size postnatally? It's mostly attributable to a very rapid initial phase of fetal-like accelerated brain size growth postnatally in the first 1.5 to 2 years. So this first 1.5 to 2 years, showing humans here, separating males and females, you see this very explosive, rapid rate of brain size growth, which is not found for as long a period of time in chimpanzees down here. And so that accounts to a large extent for how humans attain a large brain size. And all of this reorganization of the brain, again, mostly based on increasing white matter volume, which is the connectivity of the brain, is occurring in a very different postnatal social environment. It's also instructive to look at variation in brain size growth among the great apes. So I just showed you data from humans and chimpanzees, and up until very, very recently, there were no data on the rate of brain size growth postnatally in any other great ape species whatsoever. So there's no other baseline of comparison. But very recently, we've had the opportunity to work together with colleagues in Rwanda to obtain necropsy data from mountain gorillas and to be able to study the rate of brain size growth postnatally in this species. And it's a very different picture. Whereas chimpanzees achieve very close to their adult-like brain size at age 4, and they have smaller brains than gorillas, gorillas are attaining adult-like brain size about the age of 3. So a year earlier than chimpanzees, even though their brains get to be quite a lot larger. And they're able to do this in part because they rely on a stable dietary resource in the form of dense foliage that they eat, including celery and bamboo, and they have gut specializations to be able to process those sorts of food items. So they can afford, because they're surrounded by relatively low quality but abundant food that they can process, they can afford to grow quickly. But there might be some effects on social learning because of this. In gorillas who have these relatively fast-growing brains, we don't have the same kind of evidence for the range of socially transmitted behavior that one sees in the slow-growing chimpanzees. And also we know that orangutans, in terms of body size growth, are also very slow-growing. And they also have more cultural variation in their behavior. So perhaps this variation, even among great apes, reflects the capacity to extend social learning opportunities through changes in the rate and the length of brain size growth postnatally that influences the accumulation of socially learned behaviors expressed as culture, where we don't see that sort of variation in gorillas. So aside from brain size growth, other recent studies have examined other features of the human brain development in comparison to our close relatives. I'm just going to go through these very quickly. Most of these comparisons are comparisons of humans versus chimpanzees versus macaques. We have very little data from other species. So it's been shown that many brain-related genes are delayed in their expression over development in the prefrontal cortex of humans compared to other primates. And also, new genes involved in neocortical developments have been incorporated into the human genome at an accelerated rate. And one example of this is, that's just been reported, is sRGAP2, which is a recently evolved gene duplicate in humans, and experimental expression of the human-specific form of this gene in mouse pyramidal neurons extends the phase, prolongs the phase of dendritic spine development, and results ultimately in higher spine densities. So this prolongs brain growth also possibly in humans. And then there's some paleontological evidence that there are also very important changes in the proportions of parts of the brain that are enlarging early in human development within the first year, that are distinct than what's observed in chimpanzees, but also distinct compared to Neanderthals, where we have some infant and juvenile endocranial data, where there's very early parietal and cerebellar expansion that's very distinct in humans. But it's difficult to interpret those results at this point. Other comparisons of the detailed microstructure of neocortical development in humans and non-human primates are really limited, for the most part, to human macaque comparisons. And this is unfortunate because macaques are old world monkeys that have undergone independent evolution from humans for the last 30 million years. And so even if we observe a difference between humans and macaques, that can't tell us whether those specializations are unique to the human terminal lineage or might have occurred any other point in phylogeny in the last 30 million years and might also characterize apes, for example. But this leads to some important questions that we can address. So in macaque monkeys, we know something about the schedule of synaptogenesis. There's an initial overproduction of synapses which are later pruned away postnatally postnatally based on activity and experience. And you can see this here from the classic work of Pashko-Rakesh and colleagues. Across many different cortical areas, there's a synchronous peak and pruning in synapse density. So across prefrontal cortex or primary visual cortex, the timing of that peak is around the same. And it's relatively early in life. Within the first few months is the peak, and then gradual pruning and synaptic retraction. In humans, it's been argued that this is very different, the process of synaptogenesis. So here are really surprisingly, some of the only data that we have directly from human cerebral cortex for changes in synaptogenesis from Huttenlocher and Dabholkar. And here, it's data from prefrontal cortex, from auditory cortex and visual cortex, and really just a handful of subjects, 12 or 13 subjects here. And the peak in synapse density is quite a lot later, in later infancy or childhood. In prefrontal cortex, perhaps extending as late as 5 years of age. And people have interpreted these data to suggest that there's a delay in prefrontal cortex synaptogenesis as compared to other regions. But this is the data it's based on, which one would like to have higher quality. There's also data on other aspects of microstructural development that are important in establishing connectivity, which include the formation of white matter and myelination that allow us to compare macaque monkeys and humans. And so in macaques, we know that the volume of myelinated axons in the white matter increases until around the time of puberty, then stabilizes on adult-like levels. Whereas in humans, it's a more prolonged developmental trajectory. The volume of myelinated axons in the white matter increases beyond the time of puberty into the third decade of life. And you can see that here from these plots of white matter growth, where the green bar indicates continued volume increase of the white matter in these different developmental age groups. And here, even up into the age group of 22 to 32, you still see a lot of green bar for continued white matter growth. So prolonged refinement of white matter connectivity in humans. But we don't know whether that's distinct. So these comparisons between humans and macaques suggest a hypothesis that we'd like to test with data from apes, particularly that delayed and prolonged maturation of higher order cortical networks might be an important feature of human neuroplasticity, amplifying the capacity for cultural learning. So we hypothesize that the acquisition of connectional maturity occurs over a delayed time course in humans as compared to chimpanzees, who are the species where we could get the infant and juvenile brain tissue to test the idea. And these shifts in human neural development may be manifest in the timing of a range of different variables, including synaptogenesis, changes in neuronal morphology, and the accumulation of myelinated axons. So we were able to obtain, it's difficult to get these tissue samples these days, but post-mortem tissue samples of 14 infant and juvenile chimpanzees to compare to adults. And we also, because these were already chopped up by the veterinary pathologists, we couldn't do very good MRI scanning sorts of approaches. We used histological approaches, and we used comparison to other human data from the Yakovlev-Halim collection. So for synaptogenesis then in chimpanzees, does it look like macaques or does it look like humans? We approach this question using immunohistochemistry against the presynaptic protein synaptophysin, which here you can see it looks, in increasing magnification, it looks like grains of sand on the beach, but you can count this under high power in the microscope, and if you do it blind to the age and region of your subjects and you replicate it, you have some confidence that as you're counting these grains of sand, it's somewhat reliable, and this is what we had to do, of course. And these sorts of data then from the chimpanzee sample look like this. We studied 4 different cortical areas: prefrontal cortex, prestriate visual cortex, primary somatosensory cortex, and primary motor cortex. And unlike macaques, but like humans, we see that the period of synaptogenesis with the peak is relatively prolonged. Over the late infant and juvenile period, chimpanzees achieve sexual maturity around age 10, which then declines gradually. And so this is very much more human-like in its overall pattern. We weren't able to, with our sample, discern whether there was a delay in prefrontal cortex, which arguably is even hard to see with the currently available human data. We corroborated those counts from synaptophysin using electron microscopy of a subset of the sample, and this is just to show how highly correlated those two measures of synapse density were across the sample. Okay, so then we can also look at other aspects of the morphological development of cortical neurons in chimpanzees to compare to humans. And this is an image from the work of Cannell of parts of the prefrontal cortex in a 6-month-old human and in a 9-year-old human, showing that the dendritic branching architecture of neurons changes quite a lot over this period of postnatal development. And by adulthood, neurons in the prefrontal cortex down here, are much more elaborate than neurons in other regions of the cerebral cortex in humans, and this is most certainly associated with the more complex integrative capacity of these neurons. But how do they develop? There is some data on their comparative development across regions from Bob Jacobs and his colleagues, who've done a lot of the Golgi impregnation neuron tracing reconstruction work in humans. And they studied infant neurons, pyramidal neurons in humans compared to adults in these regions of primary motor, somatosensory, prestriate visual cortex, and prefrontal cortex. And the overall take-home message here is that there is a delay in the development of prefrontal cortex neurons in humans. They become more complex later. They start in infants as some of the least complex neurons in the cerebral cortex, yet by adulthood, they've overtaken all the other regions and become the most complex sorts. And so this suggests a longer time course of plasticity in the prefrontal cortex and for executive functions to be more open to these external influences over development. The data from macaque monkeys suggests that this may not be occurring with a prefrontal cortex delay in the elaboration of neuronal morphology. Here you see prefrontal cortex area TE and V1 and a number of different measures of neuron morphology of pyramidal neurons. They actually always, throughout the course of development, are more complex than in other regions. They're not developing relatively later. So we wanted to study this in the sample of chimpanzees. And using Golgi impregnation, then, we can trace neurons, map synaptic spines on the dendritic shafts. We trace them 10 neurons per region of interest so that we can obtain quantitative measures of their complexity. And so this is just example neurons from each region comparing the adult data in chimpanzees to infants. And just at a glance, you might see that it very much resembles humans. So the infant chimpanzee neurons in the prefrontal cortex are smaller than in other regions, yet by adulthood become quite a lot larger. And you can see this if you focus on the purple bar, which is the prefrontal cortex neurons, for all these different measures of neuronal complexity across development. They start relatively smaller, and then they end up quite a lot more complex. So this suggests that there is shared similarities between humans and chimpanzees in terms of the developmental time course of synaptogenesis and neuronal morphology. And this might, in part, help to explain some of those similarities that we see with the cultural capacity, in fact, of chimpanzees as compared to humans. It looks like I'm running out of time. I'm being told I have to scoot past these last two slides. So I'm going to scoot past these last two slides on myelination, which in fact do show differences between humans and chimpanzees, where humans take longer to achieve adult-like myelination, whereas chimpanzees achieve adult-like myelination by the time of puberty, in fact, well before it. And this is very interesting because human foraging skill acquisition occurs in traditional hunting and foraging societies like the Hadza and the Kun, even past the age of sexual maturity, and that's very unusual among mammals, to delay the acquisition of skilled behavior so late in development. And this is probably because of the importance of culturally acquired techniques to be able to be a skillful hunter as a human, and supported by the kind of cooperative breeding strategy we have, where there's provisioning of relatively dependent offspring as they're growing very, very slowly. So this suggests a model where brain enlargement in humans, which results in the addition of more neurons in the neocortex, as well as other regions, comes with energetic costs. To grow such a relatively large organ that is energetically expensive, paid for with a shift in diet that's been well described, with incorporating more animal resources, including cooking to make those energies more available. Along with brain size increase, the example of the agranular anterior insular cortex, certain parts of the heteromodal association cortex just predictably get larger. We also see this effect in the prefrontal cortex as well, and this may serve as a substrate for social cognition and language, providing increased computational synthesis of inputs, more capacity for executive control. This happens in association with a relatively slow altricial process of brain development, where to support these very dependent offspring that are not able to forage independently, we need cooperative breeding strategies, grandparenting, elongated lifespan for older humans to be able to contribute to the cooperative care of infants. And in that more rich social context, we have slow brain development, which can incorporate information into the production of culture. All of these things interact, of course, and ultimately, I think, contribute to what we see as elaboration of culture in humans. So I'd like to thank you all for sticking with me this late in the day. You're about to eat dinner. And I'd like to acknowledge my many collaborators, institutions that provide funding, and also the invaluable contribution of brain tissue to be able to do this sort of research. So thank you.